It is my great pleasure to introduce this afternoon's Wednesday afternoon lecture by Dr. Jean Bennett of the University of Pennsylvania. You have a page that describes some of her background. I understand there's some NIH connections in what she's going to tell you about, and that she spent a little formative time here in Building 10, which maybe she'll mention in the course of her talk, getting excited about gene therapy. Back in the days where gene therapy was a somewhat uncertain prospect. And certainly we would all agree that over the course of the last 25 years or so, the idea of gene transfer into humans for therapeutic purposes has had quite a roller coaster experience, from those heady days where it seemed as if this was going to be very easy, to the realization that delivery in an efficient way could be a huge challenge, to a recognition that the immune system had some other ideas about what to do about genes that were transferred in with various vectors, especially those involving viruses. And so it was with great delight that we saw the work of Dr. Bennett and her colleagues begin to take shape that she will tell you about today, which I think we could consider to be a true success in the gene therapy arena that is really quite stunning and dramatic in its consequences. I guess the other NIH connection I will point to is that the gene she's going to be talking about, RPE65, which when mutated causes Leber's congenital amaurosis, is a gene was identified here by an investigator in the National Eye Institute and provided the beginning of a pathway that has led now to the story she's going to tell you, traveling through the demonstration of effectiveness in a dog model and on to the application to humans, and in very significant ways, offering the chance for young individuals who were headed towards complete blindness to actually have a recovery of sight. So it is a great joy to have a chance to have a story of this sort emerge, and it's certainly wonderful for us here at NIH to have Dr. Bennett herself come and tell us about it. It's a wonderful example of the way in which basic science, carefully studied by many people over the course of multiple steps, can then lead to a therapeutic application that is clearly enormously beneficial to people with this rare disease. So please join me in welcoming Dr. Jean Bennett.
Thank you so much for such a warm welcome and such a wonderful invitation. I've had just a marvelous day so far, speaking with everybody, and have enjoyed the hospitality tremendously. It's a real pleasure to be here. Dr. Collins alluded to the fact that I'd been here in the past in Building 10, this very building, and that is indeed the case, and that was very formative for my gene therapy career. That was a collaboration with Dr. French Anderson in his laboratory more than 25 years ago, and it really was formative in terms of my career. At that point in time, the tools were not available for doing what we can do now, but French had many of the ideas that are being put into practice, and it really inspired me and in fact inspired me to go to medical school so I could learn about the diseases and then go back to the lab and try to treat them. So in any case, let me tell you a little bit about our story, and shown here is Lancelot. Lancelot was born blind with a disease which is also carried by people with a disease known as congenital blindness. He could not find his way around the animal colony to find his water bowl, to walk out of his run, to walk down the hall. He'd bump into things, and instead he became very timid and would sit down. And then 10.5 years ago, July 2000, he received an injection in one eye of a recombinant virus carrying the gene of which he's defective. Again, that gene identified here at an NEI and NIH by Michael Redman and colleagues, and it changed his life. He changed his personality, he became outgoing, went on to bigger and better things, as you can see here, reading his own journal. Had a lovely wife and sister, Guinevere, who also had the disease, and with her had multiple puppies, 6 dozen grandchildren, all of whom have been enrolled in our studies and have all benefited. So the story began with him more than 10 and a half years ago. I might add, he is doing very well. He's living at the University of Pennsylvania large animal facility in a run studded with plaques and awards, and drinks out of a silver-plated water bowl. But where are we with gene therapy? There is as yet, no approved gene therapy product, yet gene therapy research continues to go very well. And we believe, in what I want to convince you today, is that ocular gene therapy holds a lot of promise for this disease and potentially others down the road. But before I go anywhere, I would like to disclose my conflict of interest. In 2001, I filed a patent on behalf of all the collaborators shown here in all the studies involving the dogs for a method of treating or retarding the development of blindness. And this includes my husband, Albert McGuire, who is the surgeon of all the animal studies and the human studies I'm going to tell you about. In 2002, we both waived any potential financial gain. So anyway, I told you about Lancelot, and Lancelot has been to Congress several times talking about the importance of funding for biomedical research. And of course, what we realized we wanted to do after seeing all of the success with Lancelot was bring the same sort of success to a child. Shown here is Corey Haas and his parents at their visit to Congress last May. Corey Haas was born with the same disease that Lancelot has, again, severely visually impaired. And when he was 8 years old, in fact, 2 days after his 8-year-old birthday, he received a single subretinal injection, and his vision has been restored in that eye. And I'll tell you about that data. And by the way, I'm showing some images of individuals in our clinical trial. They've all gone out to the media themselves and have signed media consents. So what is the status of the genetics of retinal degenerations? And this is what allows us to begin working on these studies. As you can see here, there's been great progress in identifying the genetic bases of these various retinal degenerations. Over the past two decades, we've come to the point where we now know that there are more than 200 different genes which, when mutated, can give rise to retinal degeneration. And the vast majority of these have been cloned, and we have many excellent animal models for many of these diseases. In fact, I've indicated where the RP65 disease fit on this scale, with the first human mutation being identified in about 97, and mutation in the dog model, which is a Swedish Briard dog, identified very shortly thereafter. And simultaneous with this growth in genetics, we've developed all of the tools with which to deliver genes safely and stably to the retina, and that's about over the same time course. A lot of this coincides with development of novel viral vectors and safe viral vectors which can carry out this goal. And shown here are some of the landmarks in terms of reversing blindness in a dog model and then getting to human clinical trials. So what is this disease? Leber's congenital amaurosis is a rare autosomal recessive disease. There are estimated 4 or 5 babies born per year in the United States with this disease. And it causes very early onset vision disability. In fact, it's usually identified by the parents who realize that their 6-week-old babies aren't fixing and following the way normal babies are. And instead, they have this abnormal rotatory eye movements known as nystagmus. And the diagnostic test is electrophysiologic, looking at electroretinograms or ERGs. Normally, there's a very clear, defined waveform after flashing light to the retina, and instead, these children have flat ERGs. And no matter what parameter we look at over time, shown here is visual fields over time, whatever they have decreases, and shown here is light sensitivity. And it decreases as cells in the retina die off. This is a degenerative disease, and so there's limited time window in which one can act. The RP65 gene is produced in the retinal pigment epithelium. Shown here is a cartoon of the retina with the retinal pigment epithelium and photoreceptors. And there it forms a function which is important in recharging vitamin A so that 11-cis retinal can be produced and delivered to the photoreceptors, these cells here. 11-cis retinal is needed in order to form a light-sensitive molecule known as rhodopsin. Without that, no rhodopsin can form, and thus the photoreceptors cannot respond to light. And so the idea for gene therapy here is really quite simple. Deliver the normal copy of RP65 gene to the retinal pigment epithelium to overcome this biochemical defect. Now, how does one do that? The way we did it and our colleagues did it is using adeno-associated virus, or AAV. This is a small virus which in its wild-type form has never been shown to be pathogenic. It's never been associated with disease in humans or animals. And it carries a transgene cassette with no open viral reading frames, which is good from an immunologic perspective. What we did is use a constitutive promoter, shown here. It's a chicken beta-actin promoter with a CMV enhancer to drive the normal copy of the human RP65 cDNA, and we delivered that subretinally. Now, what is the current status of gene therapy trials? There were 3 trials that began near contemporaneously in 2008, and it was 2007, and those included one run by Children's Hospital of Philadelphia, which was our group, University of Pennsylvania, University of Florida, and University College London, Targeted Genetics. And those have proceeded so far with great results. So far, results from 19 subjects have been published, and there are probably at least 28 different subjects who have been treated in these various trials, as judged from reports on websites. But there have been a couple of additional trials added on in different sites that are phase 1 or 2. This is the clinicaltrials.gov site, which lists up-to-date clinical trials. So our trial has been completed, the first phase 1/2 trial that involved 12 patients, and I'll be telling you about the data there. We are recruiting for a follow-on study, which will inject the contralateral eye, and we are awaiting initiation of a phase 3 clinical trial with the hope of getting approval for this treatment as a drug. So what is the data that led to these studies? Shown here is results from one of Lancelot's children before treatment. You can see he's bumping around this room that is full of little obstacles. Every time he bumps, you can see there's an asterisk, and it's painful watching him walk around. Where in contrast, after treatment, the dog walks around without any problem whatsoever and avoids all the objects. There are many other outcome measures we looked at in these animals, and I'll just show you a couple more. One is pupillary light reflexes. So you're probably familiar with the fact that if you flash a light in your eye, your pupil will constrict. And that doesn't happen in dogs or humans that have this disease and are untreated. So, for example, if you look at this dog, the light is illuminating his left uninjected eye, and you can see the pupil is wide open. And then the light is shown on the contralateral eye, and you can see that the pupil constricts briskly. And this is something that it's a reflex that one can't control. We also looked at the age range for treatment success in these animals, and before I get to that, take a look at this. This shows the localization of the protein after subretinal injection. What you're seeing is scanning through the retina of an injected dog, and you can see the protein exists only in the area that it was injected. But back to the title, what is the age range for success? So we, just to summarize, we did numerous studies in dogs and mouse models of this particular disease. And what we found is that the best results were found in animals that were young, in dogs, animals that were up from about 4 months of age to about a year and a half of age. And that if we went older than that, the amount of rescue was limited. And we found very similar effects in mice. We found the best results in young, i.e. 2 to 4 month old mice, but the efficacy decreased significantly in older mice, and that's graphed out here. In the summary of the findings from the animal studies is as follows. We found that a single subretinal injection in young affected dogs or mice led to stable expression and reversal of blindness. And we showed through subjective and objective measures evidence that there was vision improvement. And we found the expression was localized specifically to the region exposed to the virus, i.e. it did not escape. And that gene transfer using AAV2 is safe. And that stemmed from numerous studies involving biodistribution and immunology and histopathology. These were all NIH-supported studies. So what to do next? How to get to clinical trial? We went to numerous people with the idea of going to clinical trial for a pediatric clinical trial for this disease based on our findings in animal models that the best effects would be found in younger animal models and thus likely younger human patients. And we went to Big Pharma thinking, well, this would be something they'd be interested in. And the universal response was, how incredible it would be to make a blind child see. How many patients? And when they would hear how few patients there are in this country with this disease, their interest level flagged. We were giving up on being able to move quickly to pediatric subjects, but in July 2005, one Dr. Catherine High knocked on my door and said, Jean, how would you like to do a gene therapy clinical trial at CHOP, the Children's Hospital of Philadelphia, aiming to treat pediatric subjects? Kathy High is the director of the Center for Cellular and Molecular Therapeutics at CHOP, and she had received funding from that hospital to establish a gene therapy and cell therapy center. She had developed a clinical grade vector core, which is run by Dr. Fraser Wright, who had previously worked at Avogen, and I hired Jennifer Wellman, who is a regulatory affairs expert, and was aware of our data and thought that this would be a great one for starters. And I, of course, said, yes, it sounds like a wonderful opportunity. And so we assembled an outstanding team of investigators, many of whom are shown here. This is the crew the evening after the first patient was injected in 2007, and then 2 years later, at the 1-year anniversary of Corey Haas's injection. It's an international team of investigators. We have a site in Italy, in Naples, Italy, that is funded by Telethon Institute of Genetics and Medicine and Second University of Naples, Sun. We have collaborators at the University of Ghent, at Harvard, and University of Iowa, and this is an ever-expanding, wonderful team. And the sponsor, obviously, is Catherine High, and the principal investigator of the study is Albert McGuire, again, who's my husband and also the retina surgeon involved in all the studies. So it's an absolutely marvelous team, and all the results I'm telling you are I am doing as a representative of this team. So what we carried out was a phase 1-2 study, which involved legally blind individuals with RP65 mutations, because obviously this treatment would not work for people with other sorts of mutations. They were 8 years old and higher, no upper age limit. They had to be able to consent and assent, or if they were children, and available for the long-term follow-up. And it was a dose escalation study with 3 doses, going from low, middle to high. We started with the lowest dose that correlates with the dose that showed efficacy in more than 90% of the dogs that we treated, and that was at the insistence of our IRB, who wanted to be sure that there was a chance of therapeutic effect given that we were enrolling a vulnerable population meaning children. This is a single subretinal injection of AAV carrying RP65 cDNA in the worst eye. And so this is how the subretinal injection is carried out, as shown here in this cartoon. It's done under direct visualization through the dilated pupil. You can see the scene here in the operating room. And it delivers the material into the subretinal space, which is an artificial space between the neural retina and the retinal pigment epithelium. And what this does is it raises a localized bleb, a localized retinal detachment in the region that the surgeon wants to target, shown here in this textbook picture. And as you can see in this particular picture, this is a picture from Corey Haas's video taken during the surgery. It's a montage of the pictures taken through the video. You can see this covered the macula. This is the optic disc here and the fovea here. And amazingly enough, this detachment flattens down really quickly within a matter of hours, leaving very little residual histologic damage, and it's very hard to find the borders later on. Now, in all of our subjects, treating the macula was the goal, because the macula is our center of fine visual discrimination. It's where we read and recognize faces and so forth. However, since patients were being enrolled at different ages and with different mutations and different extent of disease progression, some of them could not have injection in the macula. For example, this person who was 35 years old, the macula basically is non-existent. What you're looking through is the choroid, the back of the eye. So his injection was done more superiorly. These are the individuals who were enrolled in our study, as you can listed according to the dose and the extent of follow-up. We've now followed people out more than 3.4 years, with the most recent one being more than 1.5 years out. And as I mentioned, it was included an age range from age 8 all the way up to age 44, males and females. And these individuals, some of them had true null mutations, others had missense mutations. And the bottom line is there's been an excellent safety profile. There have been no severe adverse events, and I'm not even going to tell you about the majority of the adverse events, which included things like redness of the conjunctiva after post-op, which is something one would expect. There was one adverse event, which was a macular hole. Interestingly enough, this was the second patient. He had hand motion vision before treatment, which is being able to see a hand waving 1/2 meter in front of your face. And at day 14, he read 6 lines on the eye chart. And then he was obviously unaware that he had a macular hole. This had not been apparent at the time of surgery, but when we looked in through the dilated pupil, this was evident. And the surgeon suspects that this is due to a membrane which is present lying on the inner aspect of the neural retina. And so a procedure was instituted for all the other patients whereby a drop of dense oil is placed on top of the fovea, which is this very weak, vulnerable spot in the center of the retina, and to prevent that complication from happening in the future. And indeed, we have not seen that happen again. So let me show you what it was like for Corey Haas before injection. Here he is 2 days before his injection. He's walking with a cane, holding his parent's hand, walking through the hospital. And similar to the other subjects in our clinical trial, he did his homework using Braille, and then in a classroom was removed from the rest of the children, put in the back of the classroom with a video camera highlighting what the teacher was doing on the board, because they could not see what was going on in the classroom. They could not read books. And this is what Corey can do now. He's now more than 2 years post-injection. He can ride his bike around his neighborhood without anybody with him. He goes to visit his friends. He can play catch. He's a star on the Little League team in his area in New York State. Can play games with small and large objects, and many of these things that maybe his parents wouldn't want him to do. But essentially, he is leading the life of a normal 10-year-old child, and that is thanks to one single subretinal injection. So what did the subjects say? And here is just some quotes that they told us about the world is a brighter place. They can find things they drop. All in all, it has made them more independent, and the level of function that they've achieved after the injection really depends upon where they started. For example, the oldest person in our trial had a dream of being able to see her daughter hit a home run, and she did see her daughter hit several home runs. So the universal question that we're asked is, when can I have my other eye injected? Because now their worst seeing eye is their best seeing eye, and we'll get back to this topic. So let me just show you some of the results. As I mentioned to you, the first two subjects had hand motion vision, but after injection, they could read this number of letters on an eye chart. If we look at the visual acuity gains in all 12 subjects, you can see that 7 of them had statistically significant gains, and that's considered as 3 lines on an eye chart. And they have maintained these gains. Shown here are the gains in one of these particular subjects. You notice also that there's somewhat of a trend going from the oldest person enrolled in the study to the youngest person in the study, ending up with a best visual acuity. We also use pupillary light reflex. I showed you this to you with the dog model, where if you flash light in one eye, left eye, both pupils constrict, take the light away, both pupils expand, flash the light in the right eye, both pupils constrict, take it away, both expand. And you go through these cyclic changes, right eye, left eye. Our subjects before treatment had no response to light being shown in either eye. But then after injection in the right eye, in this particular subject, you can see there's no response with the left eye, but the right eye responds briskly. No response with the left eye, right eye responds briskly. So this basically converts the situation to what's called a Marcus gun pupil, i.e. one of the pupils is responding, one of the eyes is responding, and it's the eye that's been injected. And these results are stable and quantitative. We can do statistics on them in terms of the velocity and amplitude measurements. All of the subjects have gained light sensitivity, and this is a real issue for them. They all have trouble in very dim light. And shown here is the baseline light sensitivity before injection, and then in red, the sensitivity after injection. You can see there's a difference for each one of these subjects. They are ranked according to age of enrollment, and you can see that the biggest gains are found in the youngest people, as we would have predicted based on our animal studies. And shown here, there's no change in light threshold sensitivity for the uninjected eye, as we would have expected. Now, what about light levels in daily living? This is important, and this is just a preview of another test I'm going to show you. As I mentioned, all these people have trouble in dim light. Some of them don't have trouble being outside where there's very high levels of light, and some don't have too much trouble where they're in a brightly lit office area. But a room like this or the platform of the Washington, D.C. Metro would be impossible to them. So we designed another test, a mobility test, and also looked at that in different light levels of illumination. And this mobility test is shown as follows. It's a test where the subject follows arrows through a certain path, steps over objects that are jutting in the way, goes up steps and down steps, avoids black tiles, which are supposed to represent holes. We wouldn't actually want a hole there for them to get hurt with. Avoids ordinary household items like lamps, wastebaskets, ducks under a sign, and finds a door at the end. And they're graded upon their number of mistakes, or the percentage that they get correct, and the time it takes them to get through the course. And of course, the course is changed from test to test so that they don't memorize the course, because they're used to getting around by memorizing how many steps from one classroom to another, et cetera. So what you are seeing here is Corey Haas, 3 months after injection, navigating this course with his uninjected eye. His injected eye is patched, and he's immediately gone off the course. He has to be re-guided to the course, and he's saying things that are shown in print on the video. Basically, this is hard, I can't see where I'm going. He has to be coaxed, in other words. He stops here, he has to be coaxed further. He's told, just try to take a step forward, take a guess. He says, I can't even see anything. He takes a step forward and he immediately bumps into the sign because he can't see it, even though it's right in front of his face. And it takes him quite a long time to get through this course. In fact, I'm not going to show you this entire video because it takes something like 11 minutes. But simultaneous with this video, I'll show what happens when the patch is removed and put on his uninjected eye so that he is using his injected eye to get through the course. He takes every turn. He's stepping over an object jutting in his path now, going up steps, down steps, and he gets to the door without any problem. And what you don't see is that the lighting in this room is as dim as what would be found at the Washington, D.C. Metro. It's very dim. It's 10 lux. This is colorized, and the cameras are specially outfitted to be able to record in this dim illumination. So this has really maintained in a stable fashion. As you can see, he failed being able to go through the course at brighter light at baseline and at dim light, but at these various follow-up time points, more than 2 years now following the injection, he can now navigate the course with his treated eye and still can't do it with his untreated eye. And this is the same with 5 other children in this study. What about other parameters? What about visual fields? And visual fields are side vision. And visual fields are mapped out according to a grid like this. And what we found is that the visual fields expanded in each one of our patients according to the region of the retina that was injected. So this is Corey's uninjected eye, shown at baseline in blue. And then 1.5 years later, you can see in red, you can see that over time, his visual field has decreased in the untreated eye, basically because the disease has progressed. In contrast, his left eye, which was the injected eye, at baseline you can see was this blue area, and then after injection, his visual field expanded basically to reflect one-to-one the area of the retina that was injected. And just to compare, this is an older individual in our clinical trial who at baseline had this blue area of visual field in his uninjected eye that disappeared by the 1 year time point. You can see this red area here, whereas in the injected eye, which is injected peripherally, his visual field has expanded as shown by this large red island in the periphery of the visual field. Now, why is this important? And I'm going to show you how this relates to vision processing. So just as a little background, there is a one-to-one mapping between the spots in the retina and locations in the visual cortex. And this has been very well defined and worked out. And we can take advantage of this known mapping by looking with functional magnetic resonance imaging, or fMRI, to see what areas of the visual cortex are actually responding to light before and after treatment. And what I'm going to show you is some after treatment results. This work was led by Dr. Manzar Ashtari, who is the head of the DTI core at Children's Hospital of Philadelphia. And what you're going to see is the response of Corey Haas, who was injected in the left eye. You can see his left eye was flashed with light here. And you can see there's very, very little response in the back, shown in his visual cortex. In contrast, after his injected eye was flashed, you can see there's very strong response mapping to the region of the visual cortex, which maps one-to-one with the predicted region based on the injected area of his visual field. And I'd like to add that this is dim light that is flashed. This is the light that is about the level of the Washington, D.C. Metro. And then similarly, looking at an older patient who received injection more peripherally, this person was similarly flashed with light in his uninjected eye. You can see there's no response to this light, whereas his injected eye is flashed with light. And you can see that there is a very strong response that is more lateral, mapping almost directly to the area of his retina which is injected. In other words, the activation maps one-to-one with the region on the retina that is injected, and that retina is now more sensitive to light, and the brain perceives that. That is vision. So let me move on to what will be the next gene therapy study after the first set of LCA RP65 trials. So I alluded earlier to the fact that people have asked, when can I have my other eye injected? So I believe the next study will be readministration. And We did some studies prior to proposing a readministration study in large animal models because there are some very important consequences. What are the immune consequences if subjects have already received a subretinal injection of AAV in one eye or given a subretinal injection in the contralateral eye? Will they be able to gain benefit from that second eye injection, or will it hurt the initial results in the first eye? And also, what are the effects of injecting in individuals who have high serum anti-AAV antibodies in clinical trials? These people had been excluded from participation in clinical trials, and maybe they were being excluded unnecessarily. And so we did readministration studies in 2 models, both affected dogs and unaffected non-human primates. These are large animal eyes, obviously. The plan was to inject one eye first, and then 3 weeks later, when the immune response would be at its most vigorous, to inject the second eye. And incidentally, the non-human primates had been previously immunized with AAV2, so we were looking for the worst case scenario. The bottom line is there was no problem. Shown here is one of our dogs who received readministration in the second eye, and shown as another one. They both developed electroretinograms after the injection. They both can play. In fact, that's Corey playing with one of them. And in fact, we've adopted both of these dogs. The monkeys did equally well. They both showed RP65 expressed in the different eyes, although they obviously don't have the disease, so we can't look at the ramifications of disease. Two of the 4 monkeys we looked at developed T-cell responses to AAV2, but we looked at this and separated the CD4 and CD8 cells and showed that it's only the CD4 cells that show the response, i.e. it's a helper response, not a potentially harmful cytotoxic T-cell response. So that we argued, and we also looked at the possibility that people and that animals develop antibodies after intraocular injection of AAV. They do develop increased antibodies in their anterior chamber fluid. Perhaps that would hinder readministration, but that did not, according to our animal studies. And finally, we showed that even in a human study, separate from these animal studies, we showed that even individuals who have very high anti-AAV2 antibodies in their sera have very minimal antibodies in their intraocular fluid, so that it is wrong to exclude those people from participating in a clinical trial. So that has altered our inclusion criteria. So what are the implications of the results so far? I think, as you can see, that even this information has been published in the National Enquirer. The public has heard about this, is excited about the possibility of gene therapy and restoration of vision. And no, that's Jen cheated on again, not Gene treated on again. But we believe that there are many new opportunities for gene therapy for eye diseases. There's a great safety profile, great recovery in younger subjects. At least for this disease, the optic pathway is intact and can be reactivated. And this particular study could be a stepping stone towards other diseases. And I'd just like to close with some of my own thoughts about this trial, which has been an incredible experience. This has been a wonderful opportunity working in uncharted territory, allowing multiple interdisciplinary avenues of research to be carried out. And it's allowed us to change some of both the local and the broader gene therapy approvals that are going on. For example, we obtained approval to enroll pediatric subjects for this non-lethal disease, and immediately the other clinical trials that are ongoing changed their enrollment criteria lowered the age limit. So we think that that's been an altogether great, helpful change. What are some of the challenges for future ocular gene therapy trials? Well, as I mentioned earlier, there are at least 200 different diseases that can, different genes which can cause these different diseases. And it's going to be very difficult, first of all, to do all the genotyping on these patients, but also to develop a drug for every disease, and also to discriminate benign from pathologic lesions. So we've got a lot of challenges in front of us in terms of the genetics. We've got challenges in terms of patient selection and timing of therapy because, as we talked about, this is a degenerative disease. The number of cells that is available over time decreases, and so the window of opportunity will differ from disease to disease. But there are possibilities out there for restoring or adding vision to the remaining cells in the retina, which are very, very exciting. What I'm referring to is optogenetic therapy, and these results have been published recently. There are also important challenges in terms of determining what the FDA determines is clinically meaningful. So far, it's the only clinically meaningful improvement for vision research has been 3 lines on an eye chart. But what about increased peripheral vision? What about the ability to read a book or to be able to walk without navigational aids? And the ability to wake up in the morning and still see light? These are really important things that need to be discussed further with respect to different diseases. And just to summarize, this has been an incredibly rewarding experience, has presented multiple challenges with some professional risk, and I've been lucky to be a tenured professor to be able to take on this risk, and as have been the other leaders in our project. There's endless bureaucracy with a sort of hurry up and submit this and then wait. So it's frustrating in that regard. But I think the work and the success of all of the teams that have been working on LCA due to RP65 will hopefully make this easier in the future for other diseases. And finally, I'd like to point out that this is already changing the future of medicine at University of Pennsylvania, which recently built this new building called the Perelman Center for Advanced Medicine. And in the ophthalmology clinic, which is on this floor, there are 3 exam rooms devoted to gene therapy. So I'd like to thank you once again for your attention, and thank the subjects and our families and all of the people who have helped us, and acknowledge all of the many collaborators who were instrumental and totally contributed everything to this project. Thank you.
Thank you for a really exciting presentation, and we have time for questions, so there are microphones in the aisles for those who would like to address those. Let me start out by asking, do you know what the state of the AAV is in the retina, considering that it's there now for quite a long time and stable? Does it get integrated into the host chromosomes, or does it exist episomally? Do we know?
The AAV does not appear to integrate. There have been a number of animal studies that we and others have done looking for integration and have found very minimal evidence that it integrates. We believe that it stays episomal, but one of the fortunate aspects of the target organ that we're using, the retina, is that it's terminally differentiated at birth. And so these cells don't divide, and the transgene doesn't get diluted over time. And that's probably the explanation of why one single injection can allow for persistent responses over up to a decade, at least with the dog so far. But that may be more of a problem for other target organs where there is cell division.
Hi, I'm Kate Saylor, and I'm wondering how readily this technology can be applied to the other 200 genes that cause retinal degeneration, and if you have started animal studies with some of those other genes, and if that depends on whether they are extracellular products or that sort of thing.
This is a hotbed of activity. So my lab and many others around the country and the world are working on proof of concept of other inherited retinal degenerations. The ones that I think are going to be the easiest to tackle are those that are autosomal recessive, where there's a loss of function and you can deliver that function back. Each one of them is different in terms of what cell type is involved and how strong the level of expression must be and the rate of degeneration. So each one has to be tailored. I think there are definitely some that are low-hanging fruit that can be tackled very easily and quickly, and that's certainly our goal, plus the goal of many other investigators, I'm sure. There are others that are going to be more of a challenge. For example, the gain-of-function mutations are not going to be as simple to treat because there you have to think of removing a protein as well as adding one back, and the levels may be more important, and there are certain genes which present technical challenges, such, for example, some are too large to fit within the conventional AAV virus, and so strategies will have to be developed to approach those. Whether it will be possible to develop a therapy for each of those 200 diseases, it is possible. Is it practical? That's an issue that we'll have to face, and we'll also have to face the economics of it, because that's really going to be a significant challenge. However, I think there will be many others that are going to be amenable.
Related to the points that you just made, could you comment about using other approaches such as nanoparticles or zinc finger nucleases or other viral vectors?
There are so many exciting avenues being followed right now. I've spoken mainly about AAV-mediated gene transfer. There are other viral approaches which I think are going to be very useful, particularly to carry in large transgene cassettes, in large cDNAs, as well as long promoter elements which can add specificity and perhaps allow one to externally regulate the level of transgene expression. Nanoparticles, of course, would avoid use of those size limits, which are problematic for a lot of viral vectors. There's some excellent work using nanoparticles and compacted DNA to treat various retinal degenerations. Dr. Muna Nash is one leader in this area. And use of some of the other technology that you mentioned also is really exciting in terms of being able to manipulate gain-of-function disease. I think the zinc finger area is really exciting, particularly in that and other strategies get at the possibility of fixing the exact genetic problem, getting at the exact cause of the disease instead of just adding in a new gene. So those, I think, are very exciting possibilities.
Are you exploring some of those?
We are. Yeah, we are exploring a number of those. Yeah.
Gene, let me ask another question about going forward with other diseases, because you'd love to see this really dramatic success story extrapolated. Obviously, for you, having Lancelot and those other Briard dogs was an incredibly important step, because it gave you the encouragement that this was really going to work in a large animal model. But for many of those other rare diseases, maybe there isn't a large animal model, and it's a lot harder to do this in mice probably than it is in a dog. Having those shown that the strategy works for RPE65, do you think it's going to be necessary to demonstrate effectiveness in an animal model before going to humans? As long as you've got a vector you can show is safe, do you have to prove efficacy before just going to a phase 1-2 trial?
That's a question which we've been toying with and many other people have been toying with. We are lucky in this area to have a lot of really great animal models, small animal models and large animal models, but we don't have models for everything. And it certainly would be great to have large animal models for a lot of these different diseases, just because they can help you predict what would happen surgically, et cetera. The question of moving forward without proof of concept is something we've been thinking about. My prediction, I don't think that's going to happen right away, because this field is still very new. We're still cautiously moving forward, looking at safety, changing one variable at a time. I imagine the next set of studies will use different viral vectors, different genes, different targets, and so forth. And I think once there's a cumulative body, a safety body, then people will, particularly the FDA potentially, will look at the possibility of going forward without without efficacy in a more encouraging light. It should be remembered, however, that the barrier to moving to a clinical trial is generation of safety data. It's not as much efficacy data. The only issue is that a blinding disease isn't lethal. It certainly causes a lot of disability, and certainly people can be injured because of their blindness and their disability. However, I think the threshold of moving without efficacy data to human will be higher than for other diseases.
So, Jean, I just wanted to congratulate you and your colleagues for really getting IRBs to consider younger and younger patients where the prospect for success is better and better. Are there lessons that you've learned in that process that you could pass along that would help other people trying to find the way to get an IRB to actually enroll the patients in which you might see the best effect? Without using profanity or anything.
I think persistence and just, I think it's really important to listen to your gut feeling and what you feel is the right way to move forward, and hold your ground. But of course the risk is being told no, but unless you ask, you don't know. And I think there will be other obstacles. It'll be interesting to see how people navigate those.
I'm a retina specialist. I'm interested in how to prevent retina detachment after injection, because some retina injection will create a retina break that would lead to retina detachment after operation in human. How to prevent that problem?
That's a very important question, and because you're right, the concern for complications of a detachment that won't flatten down or that could lead to a tear or some other problem that would require additional surgery or worsening of the person's vision, those are real issues. And Dr. Albert McGuire has thought very, very carefully about this and developed surgery which he thinks is safe, is as safe as he can make it. The idea being that that as much control is made as possible over the surgical delivery, that it's delivered to a site which is fairly central, but depending upon the stage of the retina, and there's a gas bubble there to prevent the detachment from expanding in an unwanted direction. He has instituted procedures such as having the person be positioned immediately after surgery so that if the detachment is going to expand, it will preferentially do so in a particular direction. And essentially, the only complication to date is the one that I mentioned in the talk in terms of the macular hole in that one patient, adding the perfluorocarbon to the fovea to prevent, to buttress that and prevent that from tearing. These are real risks, and I think it will be important, assuming that this sort of this is approved ultimately, so that any retinal surgeon can use it on the appropriate patient. It will be important to have training, a training set up and guidelines to share information with surgeons so that they can avoid potential complications with this delivery. And that's something that we've been thinking about a lot.
When I had my experience testifying in front of the Senate and House Appropriations Committees trying to explain why NIH investments are a good thing, I had the chance to use those videos of Corey Haas that you saw, and this is a very compelling example for people who are wondering whether NIH is just a bunch of people playing in the lab or whether we're actually serious about trying to do something about illnesses. And that made quite a mark. And Jean, your group really is a phenomenal contribution to all of us by making those kinds of visual elements available. We have one more question.
Yeah. Thank you for your nice presentation. Is there any risk for amblyopia lazy eye after the first injection?
I'm sorry, can you say that
Is there
again?
any risk for amblyopia lazy eye after the first injection before the second?
Is there a reason for?
amblyopia.
Okay. So we're concerned about amblyopia, and that's actually one reason why we started with age 8 and above is the potential risk of amblyopia after eye surgery. For those of you who are not familiar with amblyopia, it's also known as lazy eye, and it's a condition in young children whereby if the signal from the retina to the brain is stopped at a certain point, then the eye will be ignored. And in any case, we started with age 8 and over because the risk of amblyopia after surgical procedure is very low. And we have not seen, obviously not seen this in our clinical trial subjects. We have now moved down to age 3 and over for our inclusion criteria. Again, based on risk-benefit issues, the risk of amblyopia is slightly higher, but not as high as it would be in children younger than age 3. But that is something to be concerned about and approach carefully.
Well, I think you've had a chance to hear about a very inspiring story. We'll have a reception down the hall with coffee and cookies, but let us thank our presenter one more time, Dr. Bennett. Wonderful.
Thank you.